we've developed a panel of 4 variants, each of which is in a different gene. And our test looks at all 4 variants in one go. And if a patient is positive for any one of the 4 variants, we don't expect to see anybody positive for all 4 variants. That's extremely unlikely. But if any patient is positive for any one of the 4 variants, then they fall into what we call a high-risk category. And across the board, the risk of PML, according to our study, is going to be 10 times what it would be if they tested negative. So that is quite a considerable increase in risk. The 4 variants fall into 4 genes, and the 4 genes are fall into 2 groups of different pathways. One group is involved in the complement pathway, which is clearly very important in the immune system. And the other 2 genes fall into a pathway that is less well known about, that is called the HLH, or hemophagocytic lymphohistiocytosis pathway, which also includes the macrophage activation system, or MAS. Now, what's really interesting is that since we made our discoveries and published our work, we've done more digging in the literature, and we've identified a number of case reports in the literature. And of course, because they're case reports, they're probably an under-ascertainment. There are probably more cases out there that just haven't been reported. But we've identified a number of case reports that report patients who have both HLH, which is this rare it's a hyperactivation of the immune system. It's a rare condition called HLH, and PML. And whenever you find 2 very rare conditions coexisting in the same individual, you're tempted to believe that there's a causal link, or a very strong biological link, because if you just calculate the number of individuals with HLH and the number of individuals with PML, and you multiply those 2 frequencies, you get something that's 1 in a billion. So the fact that there are individuals reported with that suggests that there's a very strong link. Furthermore, we have discovered that there have been isolated reports of mutations in one of our HLH genes that we reported that have been shown to be associated with HLH. And in particular, in one case, the specific variant we identified. So all the strands of evidence are coming together to support what we have discovered. Now, the test that we have developed is freely available through a CLIA lab. There's a very simple website. There's a form that patients can go to and fill in their details. A kit will be sent to them free of charge. It's a saliva collection kit, so there's no need for a blood draw. It's very simple. They deposit some saliva in the kit and then ship it back. Again, all free of charge. And this test is present at a website that I can describe now, or I can give you at the end of the talk. I don't know which one you prefer. It's called pmlrisktest.org. And the site is designed for patients to apply directly, but it can also be done in consultation with their physician. And hopefully what this test will do is it will allow for more careful and precise management, particularly patients with MS who are about to be started with a drug that has a PML risk. There is a hierarchy of risks for drugs that are used in multiple sclerosis. Natalizumab, known by the brand name Tysabri, has the highest risk of PML. But there are almost 100 drugs in whose drug label is mentioned a warning for PML. These are not black box warnings. The black box warnings occur on a smaller number of drugs, including Tysabri. But there's probably around 100 other drugs where there is a warning for PML in the drug label. So it's well appreciated in the community that PML is a risk factor and a consideration. One of the things that we're trying to achieve is a more precision-based medical approach to helping patients decide which drug is good for them and which drug may be risky. The previous assessments for risk of PML, or the existing assessments for risk of PML, are based on whether somebody is JCV positive or not. Clearly, as we mentioned, you have to be JCV positive to develop PML. But if you exclude everybody who's JCV positive from taking certain classes of drugs, you're excluding a lot of people. And so it's not as if being JCV positive is rare, and if you expect somebody to be negative. A very large number of people are positive. So it's a very crude test. And the other approach that people take clearly is to try and avoid the higher risk drugs, such as natalizumab. Unfortunately, after speaking to many experts in the field, speaking to clinicians, speaking to people who have done questionnaires for patients on which drug makes them feel really good and which drug is really effective, patients love natalizumab. It is still, if it didn't have the PML risk, it would be probably the drug of choice in many, many cases. So, and I'm not sure if you know about the history of natalizumab. When it was first introduced some 20 years ago, a small number of PML cases were reported in association with the drug, and the drug was immediately removed from the market. But patients were so upset and complained so much because the drug had improved their quality of life with MS so much that they campaigned to have the drug reintroduced, and it was indeed reintroduced with more safety precautions included. But the fact is that patients love this drug, and to be able to offer patients a little bit more reassurance that their risk of PML is not particularly elevated, that they're not particularly in that group that is at high risk of PML, I think is an advantage. And obviously, there are more complex questions about which drug is substituted in those small number of people, and we estimate only about 1% of people will be positive on this test. What drug could form an alternative in those cases? And I'm not an MS clinician, so it's not my field of expertise, but we have spoken again to many collaborators and experts in the field, and there are drugs that work in MS, they're good drugs and have not been associated with PML. And in particular, there's a drug called teriflunomide, which has not been associated with PML at all. We're not sure why, but there's some evidence that teriflunomide, the drug itself, the chemical, actually has some anti-JCV activity in vitro studies. So that's one possible alternative. But I think this is a sort of a new and emerging area that MS clinicians who already deal with very complex situations, they're going to have to think about this and factor into their assessment.